relationship between vocal frequency and mass. My name is Amaryllis Mara Rodriguez, and my partners are Vanessa Garcia-Clore and Abby Ragsdale.
To start off, vocalization is a crucial factor of a mammal's way of life. It is important to understand and recognize the various ranges of calls they make and why they produce those specific calls. They create these different sounds to detect predators, dictate or claim their territory, make calling, and many different ways to stay in contact with their own. Vocalization is produced by the larynx. So here in figure 1, we have an anatomy of a dog model of the throat. And we don't have the larynx labeled, but between the epiglottis and trachea is where the larynx is located. And then here in figure 2, the larynx contains tissue folds, which are also known as the vocal cords. So when air passes through these two tissue folds, they then vibrate against each other, which then produces a sound. In larger mammals, they are believed to have lower vocal frequencies because of their larger larynges and longer vocal tracts. In an article called Does Size Matter? Examining the drivers of mammalian vocalization, there's also an idea that species have a stronger sexual selection, have lower calls, which result into two things: one, larger size males, or two, the potential benefit of sounding like a larger male. Another thing considered was different forms of communication, such as ultrasound and infrasound frequencies. So in our project, we analyzed different mammal vocalizations of different body masses and measured their vocal frequencies using a sound analysis software called Audacity. Our objective was to determine if there is a correlation between the body mass of the mammal and the vocalization frequencies they create, particularly to see if the greater the mass of the mammal, the lower the frequency they make. As a result of the larger size and also larger phonatory system, mammalian vocal frequency would decrease as mass increases. So we predict that larger animals will have lower range frequencies than mammals with smaller masses. The way our hypothesis will be tested will be using various mammal vocalizations that will be analyzed in Audacity, which will then allow us to analyze the frequencies of the calls.
For our methods, we used 22 species that fell into the 3 categories of rodentia, carnivora, and ungulate. For each species, we examined the frequencies that we got from different mammal sound archive databases. And to analyze these mammal vocalizations, we used the audio editing software called Audacity. So we uploaded the vocalization and analyzed it via spectrogram. As you can see here, we have some examples of a hippo call, a coyote call, and a pocket gopher call. On the x-axis, we have hertz, and on the y-axis, we have decibels. So we went through the calls, and we measured the frequency at the loudest point in the call. So here, this is the loudest point in the call for the hippo, coyote, and pocket gopher, respectively. And we did this for 22 different species. From there, we analyzed our data by graphing it, and conducting a one-tailed t-test to determine the significance of our results. Here we have figure 1 that shows the effect of mass on mammal frequency by species. So after our audio files were collected and analyzed, there were a total of 22 different species, as you can see down here on the bottom of figure 1. Generally within these species that had the lowest masses, such as the rabbit, skunk, and mouse, frequency were significantly higher than that of other species with a greater mass. But there were some outliers. Some of these smaller species were observed to have a much lower vocal frequencies than their other low mass counterparts. A good example of this is the pocket gopher, shown here in the blue. They had a vocal frequency of 305 hertz with a mass of 0.5 pounds. In contrast, mammals of a significantly larger mass, like the hippo, cow, and giraffe, had consistently lower vocal frequencies than that of other species. Here we have figure 2 that shows the effect of mass on frequency in mammalian orders. So we separated our 22 different species into 3 different major categories, carnivora, rodentia, and ungulates. And please note that we combine Perissodactyla and Artiodactyla for the sake of clarity. And from there, we used that to determine if the category that the species fell in, carnivora, rodentia, or ungulate, played any kind of role in the relationship between mass and frequency. As you can see, ungulates, represented by the gray dots, had an average vocal frequency that was lower than that of other groups. They also had a significantly higher average mass. Within order rodentia, the masses were the lightest out of all of the groups, as you can see, represented by the orange dots, but they were highly variable, with some being significantly higher in frequency and some being significantly lower. And finally, order carnivora saw the least of variance in results, with mammals of a larger mass consistently having a lower frequency, as well as mammals with a smaller mass consistently having a higher frequency. So after our data analysis, a one-tailed t-test was conducted to determine if there was a significant relationship between mass and mammalian frequency, and our final p-value, as you can see here, was 0.100, continuing on. And given that our p-value is less than 0.05, we can conclude that there is no significant relationship between mass and frequency, and thus our original hypothesis was rejected and our null hypothesis accepted.
Based on the data we have obtained, we accept our null hypothesis. There is significant evidence to suggest there is a correlation between mammalian size and vocalization frequency. Overall, mammals with a greater size communicate with other mammals within their species with a lower frequency vocalization. This is evident in the data we correlated with the larger sized hippo, giraffe, cows, and other mammals. Hippos, for example, have a mass of 6,710 pounds on average, and from our data we found the vocalization of the hippo was 243 hertz. This indicates that due to a hippo's large size, they need their vocalizations to travel large distances and long wavelengths for their calls to be heard by other hippos. For small mammals, the same holds true, where there is a correlation between body size and their frequency vocalizations. This is revealed in the data we collected for the rabbit, skunk, mouse, and this is where their frequency was higher than that of other animals of greater mass. For example, the mouse weighed 0.04 pounds, yet had the second largest vocalization call we collected, which was 4,804 hertz. This was an overall trend for smaller mammals to have a much higher frequency than larger mammals. However, there was one animal that was an outlier, and that was the gopher, which had a vocalization frequency of 305 hertz and a mass of half a pound. This discrepancy indicates that there are more factors that contribute to vocalization frequencies than body mass alone. Our results indicate there are numerous factors that go into the frequency of mammalian calls, besides just physical characteristics of the mammal. These factors that drive evolutionary changes in the variation of vocalization frequencies include social group size, sexual selection pressure on male mating calls, environment, and body size. One factor that contributes is whether or not the species is social. Social grouped mammals tend to live in larger groups, so they need higher frequency vocalizations. It is suggested that non-social mammals have lower frequencies because they must communicate at greater distances compared to social group species. This is due to lower frequency sound waves having longer wavelengths. Therefore, these travel at greater distances throughout the environment, which is most suitable for widely distributed non-social species. An example of this from our research is the gopher, which had a vocalization frequency of 325 hertz. Gophers are non-social mammals, and therefore they need long wavelength, low frequency vocalizations to alert other gophers of nearby predators. Alarm calls are another determinant factor in the correlation between social group size and vocalization frequency. Alarm calls must be heard over low frequency surrounding noise. Because larger social group mammals use their large group size to scare off predators, larger groups utilize predator-specific calls more frequently. That means an increase in social group size leads to a higher frequency vocalization. An example of a large social group from our research is the rabbit. Rabbits are social species, and they had a higher vocalization frequency of 2,125 Hz. Even though our research was not centered around sexual selection pressures of mating calls, this was another contributing factor to vocalization frequencies. The vocalization frequencies we used were not as specific as to whether they were from a male or female. However, we know that inter- and intraspecific sexual selection are key factors to the length of vocalization frequencies and are also linked to body size. Males tend to have larger pharyngeal organs than females, and these allow males to demonstrate their social dominance within their species through communication of their deeper calls. Independent acoustic components reveal important information on their different phenotypic aspects of male quality, which are in turn important for sexual selection. This indicates that even within a species alone, there is variation in call frequencies between males and females. This portrayed an issue in our research to determine our hypothesis because mammalian calls are not gender specific, at least the ones that we found. This may have led to some discrepancies in our data. In addition to factors we cannot control in our research, such as not knowing if the call was from a male or female, there may have been possible sources of errors on how the vocalizations were analyzed. These could have been procedural errors using Audacity. An example of this could be a misinterpretation of the highest peaked frequency. This would negatively influence our data as consistency with the highest frequency peak was important in our research. This example may have been a determinant factor in how we compared our mass sizes to frequency, which may have ultimately affected how we concluded our data.